Welcome to the deep dive. Today we're venturing into, well, the surprisingly sophisticated world inside you. The flow of blood in your arteries, arterial hemodynamics.
Yeah, it sounds technical, but think of it as the hidden engineering that keeps everything running.
Exactly, a marvel, really. We'll be exploring the key ideas from 008 Arterial Hemodynamics. Now, this source, it's written for clinicians, for doctors, right?
It is, yeah. But honestly, it's packed with fundamental physics, stuff that explains how our bodies actually work on a mechanical level. Super interesting for anyone curious.
Okay, so our goal here is to pull out the most, let's say, fascinating and essential bits. Give you a clear sense of how your arteries function, but, without drowning you in medical jargon.
Right. And when you think about it, your arteries pulse something like, what, 35 million times a year?
Wow, 35 million. That kind of performance, it definitely deserves a closer look.
Absolutely. So to understand this incredible system, we'll look at the basic physical principles, things like resistance, pressure, flow, the real basics. Then we'll dig into the pulsing nature of it, all these arterial waves, and how your body adjusts things, both short-term and long-term.
And you mentioned computer modeling too.
Yeah, we'll touch on that towards the end, how advanced computing helps us sort of visualize and understand secrets we couldn't otherwise see.
Okay, great. So let's dive in. The term itself, hemodynamics. It sounds pretty scientific.
It does, but it's actually quite simple if you break it down. Hemo is blood, right?
Right.
And dynamics is about motion caused by forces. So hemodynamics is just the study of the forces moving your blood.
As opposed to just the motion itself.
Exactly. Just looking at the motion without the forces, that'd be kinematics. Hemodynamics is all about the why and how of blood movement, the forces involved.
Okay. So what's one of the first big forces or concepts we need to grasp?
Resistance. Definitely resistance. Think about pushing water through a pipe. A really narrow pipe gives more resistance than a wide one, right?
Makes sense.
So in our circulatory system, resistance, we call it R, is basically the relationship between the pressure difference that's pushing the blood, that's delta P, and the actual amount of blood flow, which is Q. So R equals P divided by Q.
Okay, R equals Barry PQ.
And where does most of this resistance actually happen in the body?
Ah, that's the interesting part. It's mainly in the smallest arteries, the ones called arterioles.
Not the big arteries we usually think of?
Nope. These tiny, tiny branches, they're the main bottleneck, the primary source of resistance for the whole system. The pressure drops quite dramatically as blood squeezes through them.
Yeah, that's why they're called resistive arteries.
You got it. Their narrowness, their length.
Uh-huh.
That's what creates most of the resistance blood encounters.
And that brings us to something called Poiseuille's law. Sounds a bit intimidating.
Huh. Yeah, it sounds complex, but the concept is straightforward. It basically just describes how all these factors connect. The flow, Q, the pressure drop,
Yeah.
the blood's viscosity or thickness,
Right.
the vessel length, L, and really importantly, the vessel radius, R. Okay, so how do they relate?
The formula is Q equals. Well, it's pi times R to the fourth divided by 8 times mi times L, all multiplied by delta. PQ equals 8P.
Whoa, okay. The radius, R, is raised to the fourth power. That seems huge.
It is. That's the key takeaway. That fourth power means even incredibly small changes in how wide a blood vessel is have a massive, disproportionate impact on how easily blood flows through it.
So, like, if a vessel tightens up just a little bit.
Exactly. Say an arteriole constricts by just 10%. Because of that R to the fourth relationship, the resistance to blood flow can jump by over 50%.
Wow, 50% from a 10% change.
Yeah. It really shows how sensitive our blood flow is. How things like, muscle tension around a vessel or signals telling it to constrict can dramatically alter resistance and ultimately blood pressure.
That really puts things into perspective. Even subtle changes matter a lot.
Absolutely. And understanding this Poiseuille flow, it also helps us understand another key concept. Wall shear stress.
Wall shear stress. Okay, another physics term. What's that about?
Think of it like friction. It's the force of the blood rubbing against the inner lining of the blood vessel. That lining's called the endothelium.
Like water flowing against a riverbank.
Exactly like that. It's the tangential force acting along the vessel wall. We call it tau. Now, it is a type of stress, but it's very different from blood pressure.
Different how? In magnitude? Direction?
Both, really. Wall shear stress is typically quite small, maybe 10 to 100 dynes per square centimeter. And it acts sideways, along the flow direction.
Okay, and blood pressure.
Blood pressure is way, way bigger, like over 100,000 dynes per square centimeter, and it acts perpendicularly, pushing outward on the vessel wall.
So one's like a gentle drag, the other's a massive outward push.
You nailed it. And we can estimate this shear stress using viscosity and
That-
flow velocity. Basically, is roughly much times the maximum velocity divided by the radius. So thicker blood or faster flow generally means higher shear stress.
And both pressure and the shear stress are important for the vessel.
Critically important. They're key factors in what we call vascular mechanobiology, basically, how these physical forces affect the health and behavior of our blood vessels and the cells within them.
Okay, so we've talked about the forces of the blood. What about the stress within the actual wall of the artery? That's where hoop stress comes in, right?
Yes, exactly. Hoop wall stress sigma hoop, is the stress acting around the circumference of the artery wall. It's caused by that internal blood pressure pushing outwards. It's a major factor determining how the vessel functions and holds its shape.
And there's a law for that, too.
Yep, Laplace's law. It says hoop stress equals pressure, P times radius, divided by the wall thickness, H, hoop, P-R-H-A.
So higher pressure or wider artery increases the stress on the wall,
Right.
but a thicker wall helps handle that stress.
Precisely. And just to give you any of the scale, hoop stress is much larger than wall shear stress, often tens of thousands of times greater. It's really the main mechanical load the artery wall has to deal with day in, day out.
And then quickly, there's also axial stress
Right.
along the length.
That's right. Axial wall stress. Sexial. It's the stress from the artery being sort of tethered or pulled along its length by the surrounding tissues. There's a formula for it, too, involving the axial force in the wall's cross-sectional area, but the key idea is that it's another stress the wall experiences.
Okay, so we have forces on the wall and stresses within the wall. How do the vessels actually respond? This is where compliance and stiffness fit in.
Exactly. Compliance C is basically a measure of how much a blood vessel stretches or expands its volume, AV, for a given change in pressure, AB or ABP, pulse pressure. C equals AVP.
So higher compliance means it's stretchier.
Right. A more compliant artery can soak up changes in blood volume without causing big swings in pressure. An interesting point, though, is that arteries actually become less compliant or stiffer at higher pressures.
And distensibility, is that similar?
Very similar, yes. Distensibility looks specifically at how much the vessel's internal area changes between the high-pressure systole and low-pressure diastole relative to its diastolic size and the pulse pressure. D equals area diastole PP. Basically, another way to look at stretchiness.
Which means stiffness is the opposite.
Pretty much. Stiffness is the material's resistance to being deformed. It's the ratio of stress to strain. Blood vessels are clever, though. They have this nonlinear behavior.
How so?
At lower pressures, stretchy fibers called elastin in the wall handle the load, so the vessel is quite compliant, low stiffness. But as pressure rises, stronger, stiffer collagen fibers start taking more of the load. This makes the vessel much stiffer at higher pressures.
Ah, a built-in safety mechanism almost.
Yeah.
Prevents them from expanding too much.
Kinda, yeah. Stops them ballooning out under pressure. Okay, so one last basic thing before we get into the pulse itself. Blood viscosity. It's not just water flowing in there.
Right, it's thicker.
Exactly. Viscosity is that internal friction within the blood, how sticky it is. Think honey versus water, right? Honey's way more viscous.
Sure.
It's due to plasma, red blood cells, proteins, all that stuff. There are ways to measure it, like with a Couette flow device, looking at the force needed to slide fluid layers past each other.
And viscosity affects flow.
Absolutely. It's a key part of the resistance in Poiseuille's law. But blood viscosity is actually quite complex. It's not constant. It tends to increase when flow is very slow, like in veins or areas where blood might pool a bit.
Oh, interesting.
And the other big factor is hematocrit, the percentage of red blood cells. More red blood cells means thicker, more viscous blood, and therefore higher resistance to flow.
Okay, got it. So we have resistance, pressure, flow, viscosity, vessel wall properties like compliance and stiffness, all setting the stage. Now let's talk about the pulse, that rhythmic nature of arterial flow.
Yes, because blood flow isn't steady, is it? The heart pumps and bursts, so we get these waves of pressure and flow moving through the arteries.
And the Windkessel model helps us understand this. Sounds German?
It is German. It means air chamber. It's a classic, simplified model, an analogy, really using electrical circuits. Think of pressure as voltage, flow as current, vascular resistance as electrical resistance, and the artery's ability to stretch its compliance as capacitance.
How does thinking of like a circuit help?
Well, the Wynne-Kessel effect explains why your blood pressure doesn't just plummet to zero between heartbeats.
Hmm.
During systole, when the heart pumps blood out, the elastic aorta and large arteries expand, storing some of that blood volume and energy like charging a capacitor.
Ah, okay.
Then during diastole, when the heart relaxes, those elastic walls recoil like the capacitor discharging, and they push that stored blood forward through the downstream resistance, the arterioles.
So the elasticity smooths out the pulsatile flow from the heart and keeps the pressure up.
Precisely. It dampens the pulsations. More advanced versions, like a three-element model, even split the resistance into proximal near the heart and distal parts. These models help us understand that diastolic pressure decay, estimate heart workload, even estimate compliance and resistance from imaging data.
Does it predict anything specific about the timing?
Yes, a fundamental prediction of the Wynne-Kessel model is that the pressure waveform should lag slightly behind the flow waveform. That's a key characteristic we see.
Interesting. Now, moving beyond that overall shape, there's this term vascular impedance. What's that?
Okay, so impedance, Z, takes it a step further. It looks at the relationship between pressure and flow, but specifically for the oscillating components, the different frequencies within each pulse cycle.
Like breaking down the pulse into its different harmonic frequencies.
Exactly. It's like analyzing the different notes in the pulse's rhythm. Impedance tells you how much resistance the system offers to flow at each specific frequency. So Z, O, kino.
And the first component, the steady part.
Right. The component at zero frequency, NO low, the average pressure divided by the average flow, that's just the regular resistance that we talked about earlier. R equals P0, Q0.
So why look at impedance at different frequencies?
Because different parts of the vascular system respond differently to different frequencies. Each vascular bed, like the arteries going to your brain or your kidneys or your arm, has its own unique input impedance. This is why if you look at flow waveforms in different arteries, they look quite different from each other. They reflect the impedance characteristics of the downstream vascular beds.
Okay, that makes sense. Different patterns reflecting different downstream conditions. And these waves, they travel at a certain speed, right? Pulse wave velocity.
Yes, pulse wave velocity, or PWV. This is crucial. It's the speed the wave travels, the pressure pulse itself, not the speed the blood is actually moving.
Right, wave speed versus fluid speed. And how fast is PWV?
Typically somewhere between 5 and 10 meters per second in the major arteries. And the key point here is that the stiffer the artery wall, the faster the pulse wave travels. Higher PWV means stiffer arteries.
How's it measured?
Clinically, a common way is to measure the time it takes for the pulse wave to get from point A to point B along an artery, say, from the carotid artery in the neck to the femoral artery in the leg, or maybe along the aorta using imaging. You measure the distance, measure the transit time, and velocity is just distance over time.
Simple enough. Is there a formula that relates PWV to the artery's properties?
There is the Mohns-Korteweg equation. It links PWV to the vessel stiffness, E, Young's modulus, wall thickness H, radius R, and blood density. It's roughly PWV equals square root of EH2R. Shows directly that higher stiffness E means higher PWV.
And as PWV, you said it indicates stiffness. Is that clinically important?
Hugely important. PWV is now seen as a really significant biomarker. It's an independent predictor of cardiovascular risk, things like heart attack and stroke, especially in people with hypertension. Because higher PWV generally reflects stiffer central arteries, like the aorta. And studies consistently show PWV increases with age, and it's higher in people with high blood pressure, particularly if it's not well controlled. It tells us about the overall health and stiffness of the major arteries.
Okay, so stiffer arteries mean faster PWV. Now, what about this idea of pressure amplification? The pulse pressure changing as it moves outwards.
Right. This is interesting. In younger, healthy people, the pulse pressure, that's the difference between systolic high and diastolic low pressure, actually tends to increase as the wave travels away from the heart, from the aorta out to peripheral arteries, like in your arm.
It gets bigger further out. Why?
It's thought to be due a couple of things. One is the tapering geometry of the aorta. Another is wave reflection. Also, the composition of the artery wall changes. There's less elastin and relatively more collagen further out, making those distal arteries naturally a bit stiffer than the central aorta.
But this changes as we get older.
It does, yes. As we age, the aorta itself, particularly the part closest to the heart, the ascending aorta, tends to get stiffer.
So the central artery stiffness catches up or even exceeds the peripheral.
Exactly. This increased central stiffness reduces and can even reverse that pulse pressure amplification. In older adults, the pressure wave in the central aorta might look very similar in amplitude or even higher than the wave measured in the arm. The amplification effect is lost.
And there's a technique called wave intensity analysis that helps explain this.
Yes, wave intensity analysis is a neat tool. It helps separate the measured pressure wave into a forward-traveling component coming from the heart and a backward-traveling component, which is the reflection coming back from the periphery.
Okay.
What happens in a stiffer aorta is that those reflected waves travel back much faster. In older individuals with stiff aortas, this returning wave tends to arrive back at the aorta during systole, when the heart is still ejecting blood.
So it adds on to the peak pressure.
Right. It bumps up the systolic pressure in the central aorta, contributing to higher central pulse pressure. But because it arrives early, it doesn't amplify the pressure further down the line in the same way it does in a younger, more elastic system.
Wow. So the timing of those reflections,
Right.
dictated by stiffness, really changes the pressure profile throughout the system.
Mm.
Okay, let's shift gears a bit. We've talked a lot about the physics. How does the body actively control all this? The short-term regulation.
Good question. Our bodies have really sophisticated rapid control systems to constantly fine-tune blood flow. They do this mainly by adjusting the contraction level of the smooth muscle cells within the walls of blood vessels, particularly the arterioles.
Changing their diameter, basically.
Exactly, changing the resistance. And these adjustments happen through two main avenues: local mechanisms right there in the tissues, and global mechanisms involving the nervous system and hormones.
Okay, what are some local examples? Intrinsic ones.
Well, there's the myogenic response. If pressure inside a small artery suddenly increases, the smooth muscle tends to contract automatically, resisting the stretch.
Interesting.
Then there's endothelial control. Remember wall shear stress. If flow increases, the shear stress on the endothelial lining cells increases, and they release nitric oxide, which is a potent vasodilator. It tells the smooth muscle to relax, opening up the vessel.
Ah, so flow itself can trigger dilation.
Yes, and metabolic factors too. If a tissue becomes more active, it uses more oxygen and produces waste products like CO2 or lactic acid. These metabolic signals can cause local vasodilation to increase blood supply to meet the demand.
Makes sense. And the global or extrinsic controls, these are body-wide.
Right. The big one is neural control via the autonomic nervous system, specifically the baroflex.
The baroflex.
Yeah, there are special stretch receptors, baroreceptors in the walls of your aorta and the carotid arteries in your neck. They constantly sense your blood pressure. They send signals up to the vasomotor center in your brainstem. If pressure goes up, the brain tells the heart to slow down a bit, contract less forcefully, and tells blood vessels to relax, vasodilate, via signals down the sympathetic and parasympathetic nerves. If pressure drops, it does the opposite, speeds up the heart, increases contractility, constricts vessels. It's a constant feedback loop trying to keep pressure stable.
A very quick moment-to-moment adjustment system.
Very quick. And then there are humoral controls hormones circulating in the blood.
Like adrenaline.
Exactly. Catecholamines like epinephrine and norepinephrine. Also, the renin-angiotensin-aldosterone system, which is crucial for long-term pressure control but has short-term effects too. Vasopressin, or antidiuretic hormone, atrial natriuretic peptide from the heart, endothelin. All these circulating substances can influence vessel tone and fluid balance rapidly.
And you mentioned earlier that the stresses themselves, hoop stress and shear stress, can trigger these.
They absolutely act as stimuli, that myogenic response. Increased pressure means increased hoop stress, triggering constriction. And the exercise example, increased flow leads to higher shear stress, triggering nitric oxide release and vasodilation. The mechanics directly influence the short-term regulation.
Okay, so that's the quick stuff. What about longer-term adaptations? How does the system change over weeks, months, years? Things like growth, aging, or maybe starting a new exercise routine.
Right. The cardiovascular system is incredibly adaptable over the long haul. It remodels itself structurally in response to sustained changes in conditions. This happens during normal growth, with aging if you become much more or less active, and importantly, in response to diseases like hypertension.
And again, those mechanical forces are key drivers.
Absolutely central. This whole area is called vascular mechanotransduction. It's how cells sense and respond to mechanical forces over time. Hoop stress, axial stress, wall shear stress, they all act as chronic signals that tell the cells in the vessel wall, smooth muscle cells, endothelial cells, fibroblasts, to change their behavior.
Like changing what proteins they make. Or growing.
Exactly. They can change their production of extracellular matrix components like collagen and elastin. They can proliferate, multiply, they can die off apoptosis, they can change shape and migrate. This remodels the vessel structure.
Let's take chronic high blood pressure again. How does that drive long-term changes via Hooke stress?
Okay, so sustained high pressure means chronically elevated hoop stress on the artery walls. This stress acts as a signal. The smooth muscle cells might start producing more collagen, making the wall thicker and stiffer. They might also proliferate, increasing the muscle mass in the wall.
So the vessel thickens and stiffens.
Generally, yes. Now, interestingly, according to Laplace's law, sup-PRH, thickening the wall, increasing H, can actually help to normalize the stress within the wall, bringing it back towards a preferred level, even if the pressure P stays high. It's an adaptation.
But that stiffening itself can cause problems, right? You mentioned a cycle earlier.
Yes, that's the downside, especially in central arteries like the aorta. While thickening might normalize local wall stress, the resulting increase in overall arterial stiffness has knock-on effects. It increases pulse wave velocity, PWV,
Which messes with wave reflections.
leading to higher central systolic pressure, which further increases the stress, potentially driving more stiffening. It can become a bit of a vicious cycle, particularly linking central artery stiffening to overall hemodynamic changes.
Okay. What about long-term changes in flow and wall shear stress?
Similar principle. If you have a situation with chronically increased blood flow through a vessel, maybe an arteriovenous fistula used for dialysis access, that leads to sustained high wall shear stress.
And the endothelium response.
Yes. The endothelial cells sense this high shear stress and, over time, signal for the vessel to remodel and enlarge its diameter. This enlargement helps bring the shear stress back down towards a normal level.
So the vessel adapts its size to the flow.
Exactly. Conversely, if flow is chronically reduced, the vessel might narrow over time. This adaptation is really important for things like the successful maturation of those dialysis fistulas.
And shear stress doesn't just affect size, but also the cells themselves, their shape.
Oh, yeah, profoundly. Endothelial cells are exquisitely sensitive to the nature of the shear stress, not just how strong it is, but also its direction and pattern.
How so?
In areas with smooth, steady, unidirectional flow and relatively high shear stress, the endothelial cells tend to elongate and align themselves perfectly with the direction of flow. They look streamlined.
Right.
But in areas where flow is disturbed, maybe it's swirling, slow, or even reversing during the pulse cycle, like often happens at artery branches or sharp curves, the shear stress is low and multidirectional. In these areas, the endothelial cells tend to look different. They become more rounded, cobblestone-like, not aligned.
And that matters because?
Well, those areas of disturbed flow and altered endothelial cell shape are known to be much more prone to developing atherosclerosis, plaque buildup.
Ah. So the local flow pattern influences cell shape, which influences disease risk.
Precisely. There are figures showing this clearly, experiments where changing flow patterns in animal arteries leads directly to these changes in cell morphology. It's thought these changes might affect how permeable the endothelium is to cholesterol or how inflammatory cells interact with the wall. It's a key link between mechanics and pathology.
Fascinating. And briefly, axial stress. Does that drive remodeling too?
It's thought to be involved, yes. Sustained changes in axial stress seem to be associated with changes in collagen synthesis, potentially leading to vessel lengthening or shortening over time, though it's perhaps less studied than hoop and shear stress effects.
Okay, so the body has all these intricate short-term and long-term ways to manage and adapt blood flow based on mechanical cues. Now let's turn to how scientists are using technology to get an even deeper look. Image-based computational analysis.
Right. This is a really exciting field that's exploded over the last couple of decades, really since the late 90s. The idea is to use medical imaging like MRI, CT scans, sometimes ultrasound, to build patient-specific computer models of arteries and then simulate the blood flow within them.
Creating a virtual replica of someone's circulation.
Essentially, yes. It allows for a personalized analysis of their hemodynamics.
What are the steps involved? How do you go from a scan to a simulation?
Well, first you need the geometry. You take the stack of images from a CT or MRI and use software to reconstruct a detailed 3D model of the arteries you're interested in.
Okay, build the pipes digitally.
Exactly. Then you need boundary conditions. You need to tell the simulation what the flow and pressure are doing at the inlets and outlets of your model. This data often comes from other measurements like specialized MRI techniques that can measure flow velocity, XMPC MRI, or Doppler ultrasound, or sometimes even data from catheterization.
So you feed in real physiological data.
Right, you use the patient's own flow or pressure data to drive the simulation. Then you use advanced computational methods, computational fluid dynamics, or CFD, to solve the equations governing fluid motion, the Navier-Stokes equations, essentially,
Yeah.
within that specific anatomical model.
Complex calculation.
Very complex. But there is sophisticated software available now, even open source tools like Vascular and Crimson, that researchers use to perform these simulations.
And what does this give you that you couldn't just see from the original scan?
It gives you incredible detail about the flow dynamics. You can get high-resolution 3D maps of blood velocity throughout the vessel, precise calculations of pressure drop across narrowings, and really importantly, detailed patterns of wall shear stress. Things you often just can't measure directly or non-invasively in a patient with that kind of resolution.
Can you give an example?
Yeah, the source material has a great example, figure 8.44. It's a case of a 9-year-old child with a condition called mid-aortic syndrome, basically a narrowing in the middle of their aorta. They built a model from a CT scan, used MRI flow data as boundary conditions, and ran a simulation. The results showed the detailed velocity patterns, the pressure drop across the narrowing, and crucially, the wall shear stress distribution. It also revealed really abnormal, disturbed flow patterns in the renal arteries, going to the kidneys, downstream of the narrowing, things like diastolic flow dominance and high-frequency fluctuations you wouldn't easily quantify otherwise.
So it helps pinpoint problems and understand the consequences of things like stenosis.
Exactly. It provides insights into the functional significance of anatomical abnormalities. It can help understand disease mechanisms, potentially plan surgeries or interventions, and even test virtual treatments or medical devices. It's a powerful way to virtually see inside the circulatory system.
It really sounds like it. An amazing blend of imaging, physics, and computing power.
It truly is.
Well, this has been an incredibly illuminating journey, really digging into the physics and physiology of our arteries. We've covered quite a lot of ground.
We certainly have. From the basics like resistance and Poiseuille's law, the nature of pulsatile flow with the Windkessel effect and PWV,
To the complex ways our bodies regulate flow, both instantly and over the long term, through mechanotransduction.
and finally touching on how these powerful computational tools are giving us unprecedented views into this system.
It really drives home how the arterial system isn't just simple plumbing. It's this dynamic, constantly adapting, incredibly sophisticated piece of biological engineering.
Couldn't have said it better myself. It's truly an engineering wonder, constantly responding to the forces within it.
So maybe a final thought for everyone listening. As you go about your day, just consider that. Consider how everything from your posture to your stress levels to what you eat to how much you move, how all these seemingly small things might be subtly influencing these fundamental biomechanical forces pulsing away inside your arteries right now.
Yeah, it definitely makes you think about the connection between lifestyle and the actual mechanics happening inside us.
It's a fascinating connection, and hopefully this deep dive has sparked a bit more curiosity about your own vascular health. Thanks for joining us.